Good morning, children. So we are back and we have already done with the Hershey Chase experiment, in which I told you that what was noticed in the medium in the first experiment and in the second experiment. In the first experiment, the bacteriophages were cultured in a medium having radioactive S35, where all the protein parts got labeled with radioactive S35, and another type of bacteria, another set of bacteriophages was cultured in a medium containing for P32 or radioactive phosphorus, which was P32, and the nucleic acid part got labeled. That means the genetic material or the DNA got labeled, and then they were allowed to infect. So when they were allowed, centrifugation was done. So what the first set in centrifuge tube, which we obtained the first set, and this was the second experiment. So there was sediment and the supernatant bacteria. Bacteria being the heavier part, it will settle as sediment along with the DNA of the bacteriophage, whereas the supernatant will have only the protein coat part of bacteriophage. It will not have the DNA part. It will not have only the protein coat part of this. So in the first case, as I told you, the S35 labeling was done, which was in the protein part of bacteriophage, the cover of bacteriophage, whereas second one, P32 labeling was done, which was in the DNA part of this bacteriophage. And when a bacteriophage infects a bacteria, what happens is the DNA goes in. That means now this DNA is not a part of bacteriophage. DNA becomes a part of bacteria. So this bacteria is in the sediment part. This bacteria is in the sediment part because the bacteriophage has injected the DNA in the bacteria. So the DNA has become a part of bacteria, and this bacteria along with the DNA of the bacteriophage forms the sediment part. Okay, forms the sediment part. So here bacteria along with this DNA, this is DNA, this is bacteria. Both DNA along with the bacteria, bacteria along with the DNA of bacteriophage, because from here to here, this has been affected by the bacteriophage from its DNA inside. So the DNA of bacteriophage now becomes the DNA of bacteria. So this all you will find in sediment, whereas only the protein coat, this protein coat you will find in supernatant. So now the protein coat is labeled with S35. This protein coat is labeled with S35, and the DNA is labeled with P32. DNA is labeled with P32. In this case, the protein coat is labeled. In this case, the DNA was labeled. There were two sets of bacteriophages. Many bacteriophages were taken here. Many bacteriophages were taken here. The first set of bacteriophage was cultured in a medium containing S35. So in these bacteriophages, a number of bacteriophages, many bacteriophages were there. Their protein code was labeled. So when they obtained this after centrifugation, this part which has bacteria as well as DNA, this was not radioactive, whereas radioactivity was detected here. Radioactivity was detected here because what was labeled in this group of bacteriophages? The protein code. And protein code was here up at the top as supernatant. So this radioactivity was detected in the supernatant. Whereas in the second case, what happened? We grew this group of bacteria in P32. And P32, that means it will label DNA. And where is DNA? DNA is inside the bacteria. And where this bacteria and DNA fuse tube, it forms the sediment. That means DNA which has P35, which is labeled by P32. So this has this. Now in this case, this showed radioactivity. Radioactivity was detected in the sediment, whereas the supernatant was not radioactive. This did not show radioactive, no radioactivity. Why no radioactivity? Because in this set of bacteriophages, the protein coat was not labeled. For this set of bacteriophages, P32 was labeled, used, and this P32 labeled DNA, not protein coat. Whereas this set of bacteriophages was cultured in a medium of S35, and this S35 labeled the protein coat. And here the protein coat which is present in supernatant was radioactive. Radioactivity was detected here in the radioactive part, in the supernatant part, whereas where the bacteria and DNA was there, it was not labeled, because we used, for the first set of experiment, we used S35. So here no radioactivity was detected. No radioactivity was detected in the sediment. Whereas in that case, radioactivity was detected in the sediment, because sediment had DNA, and DNA was labeled with P32. Is this experiment of Hershey and Chase clear to you, children, or shall I repeat this? I want your response. Is this experiment of Hershey and Chase clear to you, or shall I repeat this? I want your response. Yes, beta? Okay. Yes, it's clear. Yes. Yes, anybody who wants me to repeat it? Chitra is also saying. Anybody who wants me to repeat it just now, you people say who wants me to repeat this experiment? Hershey and Chase, very important from the examination point of view. Very important, molecular basis of inheritance. Yes? Anybody who wants me to repeat this experiment? Yes, bache? I'm not getting any response. Yes, Simran Sogi? Repetition is required. Amit Katoch? Amit Katoch, repetition is required? Clear. Okay, fine. Vanshika Singh? Aditi Gurang? Gunjan? Chambial? Yes? Girish, if you are there? Yes. So the children are saying that repetition is not required. I'll move on to the next, which is DNA versus RNA we have done. We come to the replication now. A replication. Now, replication in case of DNA is semi-conservative. Replication in case of DNA is semi-conservative. Now, what is this semi-conservative mode of replication of DNA? First of all, this we discussed that which can be for genetic material. What can be considered as the first genetic material? Whether it is RNA or it is DNA, which can be considered as the first genetic material. So RNA can be considered as the first genetic material. RNA can be considered as the first genetic material, and the reason and the justification that we can say that RNA is a genetic material is that this RNA is considered to be highly reactive. This is reactive. This is unstable, reactive and unstable. So something which is reactive, that means, and unstable, it shows evolution. This is the first region, that is, it evolved. Being unstable and being reactive, it changed. First it changed, modified itself to become stable, and then evolved as DNA. Because it was RNA which was unstable, it was RNA which was reactive. So what happened, that it tried to see, everything in the nature wants to be stable. So it tried to be stable, and for that it changed itself. It was reactive, so it changed, it modified itself to become stable, and similarly in the direction of becoming stable, it evolved itself and got converted into DNA. So it was DNA which was evolved from RNA. That means RNA got converted into DNA in due course of time, in the path of being stable. This is what changes were seen in RNA, and then it became DNA. Now this RNA, you know the difference between RNA and DNA. The main difference between the two is the RNA has ribose sugar. Ribose sugar has one extra OH, which is attached to a functional group hydroxyl, which is attached to the second carbon in the ribose sugar, whereas in the second carbon DNA has H, whereas RNA has OH. And the second thing is the presence of uracil. Uracil is present in RNA, whereas uracil is replaced by thymine in case of DNA. So this is the main difference between DNA and RNA, and maybe it is the way it modified, that it got rid of its hydroxyl group and it got rid of its uracil, which is another unstable component. As I told you in the last lecture, how this uracil is formed. Uracil is formed by deamination of cytosine. So whatever number of cytosine is present, or slowly this cytosine, some amount of cytosines gets deaminated. That means from the cytosine, which is a nitrogenous compound, the amino groups gets detached and it gets converted into uracil. Now this one more very important thing which I want to tell you is replication. Now this DNA undergoes a process, and which is a very important process. This process is known as process of replication. A replication in DNA is a very important process to make multiple copies, or to increase in amount, or to amplify. Now this replication is DNA is considered to be semi-conservative mode. Semi-conservative mode of replication. Semi-conservative mode of replication. Now, the literal meaning of semi-conservative. Semi means half. Half is conserved and half is formed new. That means when the replication takes place, half which is already there. Half from the old one is used. Old form. Half is old one, okay, old strand or old one, and half is formed new. This is known as semi-conservative mode of replication. Now as you all know, both the strands of DNA, they are helically coiled around each other. As I told you, the DNA strands are like this. They are helically coiled around each other. Now what happens at the time of replication? At the time of replication, the unwinding takes place. Already, I'm making the old strand with black color and the new strand with red color. Okay, now what happens? At the time of replication of DNA, both the strands, they open up. Suppose these are the open strands. From here they have opened. The detachment. From here they have opened up. So little they are still with each other. And from here they have opened up. So both the, winding around with each other, it has opened up here. Now what happens? This black color strand is already this and this strand. This strand is, and this strand is these black colored strand. And on these black colored old strands, the new strands are formed. Suppose here, the new strands are formed on these black. And then they also wind around them. These are not formed like this. They also wind around them. So the red ones are the new strands which are formed. So now what has happened? Now in this DNA, after replication, one strand which is already there is the old strand, and this red colored one which is newly synthesized is the new strand. So we have an old strand and a new strand. And this type of replication in DNA is known as semi-conservative mode of replication, in which a new strand is formed on the old template strand. That is, this is the template strand. That means the basis, which forms the basis for the synthesis of new strand. This template strand is the old strand, and the red colored new strand is the new strand. So we get a semi-conservative mode of replication in which half of the strand formed is new and half of the strand formed is old. Now what will happen? Now this also wants to replicate, so this will also open up. This will also open up like this. So what, when this and this wants to replicate, then what we will have? We will have again, I'll take only one, I'll take this one. So we will have now two strands. When they see, we have now two strands, one red which is shown here and another black which is shown here. So now if they want to replicate, we will have it like this. Now as a result of replication, new strand will be synthesized, which I'm making with the green color. So these are the new strands which are synthesized over the old strands. So this is again the semi-conservative mode of replication, when half of the strand is conserved and half the strand is formed new. So that is why the replication in case of DNA is known as semi-conservative mode of replication. Is it clear, children? Semi-conservative mode of replication to you? This was just an introduction of that. Is it clear? Or anybody wants repetition of this? Yes, beta? Although I use different colors, but I think the different colors are not visible on the screen. Although I have used this red and black. Either I'll have to make it little darker and thicker, then only it will be visible maybe. I'm making it little thicker, I think. This way it will be visible that this is red color. I made it little thicker so that it is visible to you. Red color, and here the new strand which is formed of the green color. I'm making this also thicker. So that the different colors are visible to you. This is red color, which is the old strand. So I think it is little visible to you. So bache, is it clear to you now? The semi-conservative mode of replication is clear, bache? Or any repetition is required? Okay, Paras is saying yes. Yes, beta. Clear. Sukh Sahaj is also saying Amit Katol. Repeat once. Okay. Replication in DNA is semi-conservative. Semi-conservative means half is conserved and half is formed new. When the DNA as you know, DNA is a double helical structure. It has two strands. DNA has two strands, and both the strands are coiled around each other like this. If you see this structure, I have made DNA. This is one strand. This is another strand which is coiled around each other. DNA consists of two strands which are coiled around each other. Now, if replication takes place in DNA, how will it replicate? For replicating, the first of all, these two strands where they are coiling, they will uncoil. That means they will open up. So if you see here, this is a old strand in which both the strands I have made with black color. So if you see here, I have made here opened up black strands. See, the black strands have opened up. The black strands have opened up means where there was coiling, that coiling has, it has opened up from one end. Say from this end till here, it has opened up. See, from here, from the last till here, it has opened up. So here the DNA strands have opened up, and each strand which has now separated. See, this strand and this strand both have separated. One is this strand, one is this strand. From down, the opening takes place. So they open up, and when they open up like this, the already the black colored strand, which were the strand from here, when they open up, these are the old strands. So on these black colored old strands, new strands are synthesized. See, the new strands have been synthesized. The red colored are the new strands which are synthesized on the black colored old strands. So the black colored old strand is the conserved strand, and the red colored strands which are the strands which are formed new. Half strand is conserved, half is formed new. So we call it semi-conservative mode of replication. Now more replication has taken place. Now if they want to replicate more, now if this strand and this strand, both strands want to replicate, again they will open up. Now when they open up from here, one strand will be red, another strand will be black. Now here also, if they open up, one strand will be red, another strand will be black. Now this black strand is the strand which has come from here. The red strand is the new strand which was formed on the black strand. Now more replication has to take place, so it will open up. Now here I have made just one strand, this strand. When this red and black strand it open up, so it will open up from down. Say from here till here it has opened up, and here still the coiling is there. Here still the coiling is there, but now it has opened up. So see, the one red strand gone here, one black strand is here. Now on this one red and one black strand, the new strand will be synthesized, which is the green one. This, the new strand has been synthesized. So again, it is a semi-conservative mode of replication, because half the strand is already existing one, or the template strand is the already existing old strand, whereas the green strand is the one which is newly synthesized. So replication of DNA can be considered as semi-conservative mode of replication. Is it clear now, beta? Or and more repetition is required? Is it clear, or more repetition is required? Yes, bache? Clear now. Okay, now we come to an experimental proof of this. Now there were two scientists. The names were Messelson and Stahel. The full name were Matthew Messelson and Franklin Stahel. There were two researchers, and the name were Matthew Messelson and Franklin Stahel. This experiment is also very important, and it was given by these two researchers, Matthew Messelson. The name of the researchers were Matthew Messelson. Matthew Messelson and Franklin Stahel. These were the two researchers who experimentally proved that replication of DNA is semi-conservative. They proved experimentally that DNA replication or replication of DNA is semi-conservative. That means they proved experiment that when the DNA replicates, half of the strand is already existing one, or the old strand is found. So they proved it experimentally that DNA replication is semi-conservative mode. Now for that, what they did? For that, they took the bacteria E. coli and they grew it in a medium of NH4Cl, where the nitrogen, heavy isotope of nitrogen was taken, and it was N15. They took E. coli. E. coli is Escherichia coli, which is many a times used in the biotechnological experiments also, and in case of the genetics experiments also. So they took E. coli and they grew it, they cultured it, or they grew it, cultured it in a medium of NH4Cl. But what was special about this NH4Cl? The special thing about NH4Cl was this nitrogen, which was used in making the solution of NH4Cl. It was a heavy isotope of nitrogen, N15. What is the normal isotope? Normal nitrogen? Normal form of nitrogen is N14. But they use this heavy isotope of nitrogen. And this heavy isotope of nitrogen was N15. So they did not use any radioactive isotope. In this experiment, what was used was the heavy isotope of nitrogen. And like in radioactivity, the radioactive substance can be detected by some, by exposing to some light, the radioactive substance can be detected. But this heavy isotope of nitrogen can be detected only by centrifugation. Only by centrifugation, the heavy isotope of nitrogen can be detected. If it is centrifuged, if both are centrifuged, say N15 and N14, both are centrifuged. You know what is centrifugation? Centrifugation means you take the material in test tubes, small centrifuge test tube. Suppose in this small test tube you have taken N15, and in this another test tube, centrifuge tubes. These are not the ordinary test tubes. These are centrifuge tubes or centrifugation tubes. In this test tube you take N15. This is A and B. And if this A having N15 and B having N14, if you take these heavy isotope and normal isotope of nitrogen in centrifuge tube and you centrifuge it, what happens during centrifugation? There is a machine, and I'm showing you the top view. If this is the machine, I'm showing the top view of the machine. The machine from the top looks like this, a circular machine. It is kept in the chemistry lab also in our school, you must have seen. And it has a number of holes in here, and in these holes you can insert these test tubes. These test tubes can be inserted in these holes. So suppose you have kept inserted A test tube has 15 and B test tube has N14, and you centrifuge it, and you centrifuge it, CENTRIFUG, you centrifuge it. Centrifuge means that it is rotated. You switch on the button and it will start rotating. The whole structure will start rotating in the direction, and the rotations are very fast. That means about 1000 rotations per minute, or say 1600 rotations per minute, 2500 rotations per minute in 1 minute. Okay, they rotate very fast. So the rotation is very fast, and when it rotates inside this centrifuge machine, what happens? The heavier substances in the test tube settles at the bottom, and the lighter substances, they remain at the top. Like I told you in Harsha and Say's experiment, that the heavier one, they form the sediment, and the lighter one, they form the supernatant. Same thing happens. Now, if this was taken in both the test tubes with cesium chloride. Here in this case, cesium chloride was used along with this. Cesium chloride, CsCl2. This was used. So what was found that the N15, it was heavier than this. It settled here at the bottom. It settled here, whereas N14 in the centrifuge tube settled here, being lighter. So this was the settlement of the heavy isotope, the heavy isotope of nitrogen, which was heavier in the which has more density. It settled at the bottom of the centrifuge tube, whereas N14, which was a normal isotope of nitrogen, it was lighter than N15, so it settled somewhere here. So this was the basis which was used by the experimental proof, which was given by Messelson and Stahel. And this Messelson and Stahel, what else they did? This I'm going to tell you tomorrow, because now 2 minutes are left, and if I start with this, I'll not be able to finish it right now. So the experiment I'll start tomorrow. Right now, I told you the process of centrifugation. What happens in the centrifugation? It is rotated at a higher speed, at a very fast speed in the centrifuge machine. When the heavier substances, they settle to the bottom, and the lighter substances, they become the supernatant. So same thing happened here when this was a centrifuge using the solution of cesium chloride. The N15, which was the heavier isotope of nitrogen, settled at the bottom of the centrifuge tube, whereas N14, which was a normal isotope of nitrogen, it did not settle at the bottom, little higher in position, so little on the lighter side. So this happened, and this experiment, it told how what is the experimental proof. They cultured the E. coli in the medium of N15. They first of all, they cultured it using the medium N15 only. All a group of a colony of E. coli was taken. We speak in the terms of colony. A colony of E. coli was taken and cultured in a medium having N14, and they were cultured in a medium having in N15 till the time that both the strands of DNA got N15 in it. That means when they will cultured for one generation, what will happen? The N15 will come onto one strand only, because replication only once will take place. So one strand will get labeled with N15. But same thing happened here. What happens here? Again, next application, they cultured that E. coli in the medium of N15 till both the strands of its DNA got labeled with N15 for 1, 2, 3 generation, till both the strands of its DNA